Today we are going to discuss about type of fixation. So the fixative can be classified on the basis of following criteria based on the nature of fixation, chemical properties, component presence, action on tissue protein. Then we will move nature of fixation. Again we can divide into subcategories. Immersion fixation, that means nature of fixation. According to the nature of fixation we can divide subcategories: immersion fixation, coating fixation, vapor fixation, perfusion fixation, freeze drying, microwave fixation. So we will discuss later one by one. Then immersion fixation, when we consider the immersion fixation, commonest way to fixation. So this is commonest way of fixation in the laboratory. In this technique, the whole specimen is immersed in the liquid fixative, such as the tissue samples are immersed in 10% neutral buffered formalin or cytology specimen in 95% ethyl alcohol. Coating fixation, this is commonly used in the cytology samples. The spray fixative is used for easy transformation of the slide. The main advantage of the spray fixatives are fixation of the cells to impart a protective coating over the smear. No need to carry liquid fixative in bottle or the jar. So the spray over the smear should be smooth and steady, and the optimal distance of 10 to 12 inches should be maintained between the nozzles of the spray and the smear. The spray fixative usually consists of alcohol and the wax. Therefore, these wax should be removed before the staining procedure. Vapor fixation. In this type of fixation, the vapor of chemical is used to fix either a smear or the tissue section. The commonly used chemical for vapor fixation are formaldehyde, osmium tetroxide, butyraldehyde, ethyl alcohol. The vapor converts the soluble material to insoluble material, and these materials are retained when the smear come in contact with the liquid solution. Perfusion fixation. This is mainly used in research purpose. In this technique, the fixative solution is infused in the arterial system of the animal, and the whole animal is fixed. The organs such as brain or spinal cord also can be fixed by perfusion fixation. Freeze drying. In this technique, the tissue is cut into thin sections and then rapidly frozen into a very low temperature. Subsequently, the ice within the tissue is removed with the help of vacuum chamber in highest temperature, that means minus 30 Celsius. So the freeze dry technique is useful mainly to study the soluble material and very small molecules. Advantages of freeze drying method: excellent for enzyme study, no changes in protein, no shrinkage of tissue, preservation of glycogen. Then immersion fixation, where When we consider the immersion fixation, commonest way to fixation. So this is commonest way of fixation in the laboratory. In this technique, the whole specimen is immersed in the liquid fixative, such as the tissue samples are immersed in 10% natural neutral buffered formalin or cytology specimen in 95% ethyl alcohol. So, commonly used fluid in the cleansing bath, liquid nitrogen, propane, isopropane. Then we will move into microwave fixation. So, the microwave fixation, normally here we use microwave ovens. So, here microwave, we all know this is the type of electromagnetic wave. Frequency between 300 megahertz to 300 gigahertz. Wavelength varies from centimeter to nanometer. A scientific microwave oven operate with a frequency of 2.45 gigahertz. Medical microwave oven operate with a frequency of 0.915 gigahertz. So basic principle of the microwave fixation, the electric magnetic field is created by the microwave, and the dipolar molecules such as the water rapidly oscillate in this electromagnetic field. This rapid kinetic motion of these molecules generate uniform heat. The generated heat accelerate the fixation and also other steps of tissue processing. The most important characteristic of microwave heat generation is homogeneous increase of temperature within the tissue, and every part of the tissue is heated. The factors controlling the temperature rising. So here we are using temperature, so we have to maintain the temperature. Otherwise, the temperature rise of the heated media depending mainly the dielectric property of the media, thermal property of the material, radiation, orientation of shape of the objective. So advantages of this method, the main advantages of microwave fixations are rapid process, no change in volume of tissue, and preservation of the tissue antigen, and good for the immunohistochemistry, and it facilitates the stain reaction without any bad effect. Disadvantages: the tissue immersed in formalin during microwave fixation may generate large amount of toxic gas. Therefore, overheat hood is required for the removal of these toxic substances from the microwave. Chances of heat injury. The applications of microwave fixation in routine surgical pathology laboratory is used in microwave fixation. The electron microscopy after osmium tetroxide fixation, urgent process of biopsies, like a kidney. So chemical properties. This is the other method of classification of fixative. So here aldehyde, normally we use formaldehyde, glutaraldehyde, oxidizing agents. So according to the fixative, according to the chemical features, we can divide the oxidizing agent, osmium tetroxide. Other one, protein denaturing agent, example ethyl alcohol, methyl alcohol. The one, cross-link agent, carbodiamide. Other one, miscellaneous pictic acids. So this is the method, the how the fixation is happen. Though we can divide into two, the physical method and the chemical method. And the physical method we can do two types, heating method and freezing method. And the heating method we can apply for the cell smears, accelerate the chemical fixation. So other one is the freezing technique. Under physical method, applications dedicate antigen non-suitable for the host protocols. So in this condition we are using freezing method. So because when we are using heat, delicate antigens are present, so when we are heating it may modify the structure. Other one is the microwave method and the physical method. So this is application primarily microwave stabilization, microwave association, fixation here we are using. In the chemical method, we can divide into three, coagulant fixative, unclear, and the cross-linked fixative. Coagulant fixative, we can use alcohol, acetone, acetic acid, pictrate. Then unclear, mercuric chloride we are using, unclear chemical method. Other one, cross-linked fixative, here we are using aldehyde, oxidizing agent. For example of oxidizing agent, osmium tetroxide, potassium dichromate, potassium permanganate, and chromic acid. Then chemical fixative, again we can divide into two categories, simple fixative and compound fixative. Simple fixative and simple fixative, we can use formalin, mercury chloride, osmic acid, pictic acid, acetone, ethyl alcohol, osmium tetroxide, and osmic acid. Here only one fixative we are using. But when we consider compound fixative, again the compound fixative we can divide into three categories, microanatomical, cytological, and histochemical. And microanatomical we are using to maintain the microanatomical structure. Here we are using formal saline, neutral buffer formalin, Sanger's fluid, and Buin fluid. Then under cytological fixative, for the nuclear fixation, we can use Carnot's fluid. Cytoplasmic fixative, we can use Campi fluid. For the histochemical, we can use cool acetone or ethanol. Then we will move the next classification of fixative according to the component present. The simple, and we already discussed compound. Simple, when we consider the simple fixative, only one chemical is present, formaldehyde, ethyl alcohol, glutaraldehyde, picric acid, osmium tetroxide. So these are the chemicals we can use only single. That means if we take the formaldehyde, it's fix the tissues when formaldehyde are present only. But when we consider the compound, fixative, this is more than one chemical is present. So here we are using more than one chemical. By using that we are preparing the solution of fluid. The example, carnoid solution. Then according to the action, so according to the action on protein, the fixative act on the protein. So there are two types, coagulative and non-coagulative. When we consider the coagulative fixative, here examples are ethyl alcohol and pectic acid. So these act on the protein or amino acid or side chains and it's produce coagulation. But when we consider the non-coagulative, example of fixative, the formaldehyde, osmium tetroxide and glutaraldehyde. So here it will react with the fixative, it will react with the protein parts, the amino acid, especially the side chains. But it's not produce a coagulation. So we will discuss the detail how the fixatives act on the protein and how is produced coagulation or non-coagulation in later. So essential precautions for the fixation. The certain essential precautions are necessary for proper fixation. The tissue should be free from excessive blood before putting it into the fixative. Tissue should be tinily cut into 3 to 5 millimeter thickness. The amount of fixative fluid should be 20 times more than the volume of the tissue. The tissue with fixative should be in a tightly screwed cap bottle. So these are the essential precautions when we are fixing or using fixative for the tissues. So these are the references. Thank you.